Based on our surveys, after I joined at ICR Directorate of Floriculture Research, my colleagues are conducting extensive surveys to document as to like what is the range of spread or the extent of spread of this phytoplasma in most of the nursery hubs where we are working. And most of the ornamentals, they are of belong to Asteraceae, and the phytoplasma that is dominating is the Phytoplasma asteraceae. In case of we are also now observing in Pune that what is the level of manifestation in different varieties in a particular species. In case of China aster, there are differences in terms of the varieties that we are evaluating. There is a clear sign of where some of them are highly susceptible, some are less susceptible. The same is the case in case of other crops, which I am going to share. In case of Dimorphotheca, we have documented all the manifestations that take place both in vegetative as well as the floral stage. This is a typical symptomology in case of Saponaria, one of the ornamental potted plant, and Dianthus, again a seasonal annual, which is very popular across the country. And then we have embarked onto the characterization with the help of the latest molecular tools to establish their the nature of the the molecular nature of the infections. In case of Gerbera, this is a typical manifestation that that is observed in case of the polyhouse grown Gerberas. It turns completely green, and it is 16 SR DNA nested piece. Through that, we could establish which is the major phytoplasma that causes this kind of manifestations in case of Gerbera. In case of annual chrysanthemum, this is the typical symptoms that we have observed in many of the annual chrysanthemum growing areas, mostly in the western part of India. And these particular manifestations I have not seen in case of Delhi when I was working with annual chrysanthemums there. What I could perceive is that the temperatures play a major role. In chrysanthemum as well, when we are growing these chrysanthemums during the peak winter, there are no symptoms of this phytoplasma manifestation. But we had some varieties which are thermo and photoinsensitive, which used to bloom in summer. In those varieties which used to bloom in summer, we used to get a lot of symptoms which are like this. Here in Maharashtra, we are observing this particular symptoms in case of China aster very often, and even marigold. So what we found is it is 100% similar to Stevia rebundina phytoplasma, which was established through the using the molecular approaches. In case of Gerbera, marigold, and annual chrysanthemum, we have found out that which is the major group that is causing the infection. That is found to be 16 SR group 1 as well as group 2. In China aster, this is confirmed again to RFLP that this is the species of candidatus. And when you look at if you grouping of these annual chrysanthemum, marigold, and then China aster and chrysanthemum, the phytoplasma that is causing is slightly different, and which is documented here. We in the morning, Dr. Ahmed sir has given an elaborate presentation on the vectors, and this is only a kind of a confirmation as to, in case of what we have observed very closely is in case of China asters, we have noticed the following species of the hoppers in marigold and chrysanthemum. In case of China aster marigold, we found that Hishimonas was the species, and Orvisius was the species in case of chrysanthemum. And we have also studied the correlation between the incidence of the manifestation appearance as well as the in comparison to the emergence of these vectors in different months. And we have also studied the role of Parthenium, which is acting as a reservoir probably, or a passive reservoir, which is contributing to the further spread of this phytoplasma after the season is over. And the Cuscuta is also being investigated, where our colleagues have established that Cuscuta is also playing an important role in transmission of the phytoplasma. Management, we are trying to do different approaches, as we all have discussed in the previous presentations. We are screening the existing varieties to see what is the level of incidence in these varieties. We have about 150 chrysanthemum varieties with us, and we have equal number of roses and equal number of gladiolus, and we are very closely observing the kind of manifestations that are being observed in different varieties. We're also trying to establish this infected material through tissue culture in order to maintain them and to investigate further for further analysis. And we have also tried the hot water treatment, which is one of the time-tested old method of getting rid of the infections, but we are not successful so far. And so far, the only silver lining is the dipping in tetracycline at 200 ppm is good when you are starting propagating your material before you establish your chrysanthemum plots. We are also trying to use different molecules which are documented in the literature by applying them at the different stages of the crop growth in order to either to revert or to minimize the symptoms. But so far, our efforts are not fruitful. As I mentioned, when we were discussing these issues with Dr. Krishna Kumar sir while he was the DDG, he has encouraged us to organize a awareness program in the nursery belt, the nursery hub of Andhra Pradesh, which is Kadiyam, where there are about 3,000 nurseries which propagate different plant material and they export even to other countries. And there is a greater requirement of this awareness. Therefore, we have organized in 2016. At the same time, we have also brought out a base paper because of his encouragement. We brought out this specific booklet, which I can share with the organizers, which can be sent to all the participants for the benefit of. So to conclude, as I mentioned, I'm not a pathologist or an entomologist, but I was drawn into this primarily because of the problems that I faced while breeding chrysanthemums. So this has intrigued me to work towards the management as well as the manifestations, and to educate the farmers so that they do not commit the mistake of propagating the wrong material and spreading the disease further. So to conclude, as I mentioned, that we need to have certified plant material from the nurseries. Awareness is also very important for the people to not to take it forward, and vector management, as rightly said by Dr. G.P. Rao as well as Dr. Hegde, and finding these solutions durable solutions through breeding programs. But it is a long rope to take, as Krishna Kumar sir has rightly mentioned, that we do not know the source from where the resistance can be isolated. And using the latest technology, maybe the gas or lateral flow diagnostics would be able to help us in future to come out with some solutions to overcome this particular problem. And in case of ornamental crops, because it is a short duration crop, and then the symptoms they appear at the flowering stage mostly. Therefore, the impact on floriculture crops is very damaging, because at the time of flowering only the marketability of the spotted plants or the flowering plants is ensured. So no doubt in any other crop, even the plantations or other crops, the economic produce, if it is affected, is going to be, hinder the growth of farming community. So this is what I wanted to convey to all of you. Thank you very much for the time that is given, and I acknowledge the contributions in our work from Dr. G.P. Rao. We contacted Dr. G.P. Rao. He was the first person to whom we contacted when we started seeing the reversion of the the most hopeful green mutant that we saw. So he was the person who has seen and then confirmed that it is a manifestation of. And then Dr. Krishna Kumar, who always was the force behind us to encourage and to work on this, because he strongly believes, and it is a fact as well, that floriculture crops do contribute for many of the biosecurity threats that we are discussing. And my colleagues, Dr. Prabha, Nitika, and Girish, they are the people who are working and then documenting this. And I'm only able to present to you the work that they have done. And also one of my students, Aido, she was the first person to work on this from floricultural group at IRA with the support and help of Dr. G.P. Rao. And she has brought out very good publications in the international journals. So thank you very much, sir, for your invitation to join this group, and it was very, very educating for me also, because I got exposure to many aspects that were discussed in the previous presentations. Thank you so much. Thanks for the organizers, and also to Krishna Kumar sir. Thank you so much.